so done.
Okay, our last speaker for the session is Dr. Marielena Sosa from Stanford University. And take it away. Oh, wait, you don't need this.
All right, thank you, Alina, for the introduction. Thank you so much to Kara for this amazing opportunity. It's really been an honor and a privilege to be here. My name is Marielena Sosa. I'm a postdoc at Stanford with Lisa Giacomo. And I'd like to start with an example to illustrate the overall research question that I'm trying to address. This is a neighborhood near my home in downtown Palo Alto. And as I walk down this street, I know from experience that my favorite patisserie is located here. And so as I approach the street corner, I tend to have a sense of anticipation about the delicious pastry or coffee that I'm going to get. And as I leave this location, I have a rich memory of that rewarding experience. And as I continue down this street, previously these businesses were closed, so this location didn't have any specific importance to me. But recently, this adorable patio caught my eye, and I realized that there was another pastry shop that had just opened here that also has delicious food. So now my experience of passing these two locations is extremely similar in its reward quality, even though they're in different spatial locations, surrounded by different environmental stimuli from the different city blocks. So my question is, how does the brain layer this information about rewards and the saliency of our experiences and our memories on top of a stable representation of the external world? And how does it do this flexibly when we need to learn new information? So the brain structure that we can look at to answer those questions is the hippocampus, which is located here in a sagittal view of the rodent brain. And pyramidal neurons in this region are known as place cells because they'll fire in specific locations in space as an animal moves through an environment, creating a sequence of activity that we refer to as the neural map. In a different environment, such as a different city block, individual cells will change their preferred firing locations or remap, and additional cells will also come online such that there's a unique neural code for this experience. But really interestingly, we see this kind of sequential activity not just during spatial navigation, but also during all kinds of tasks. So for example, you could have an olfactory cue followed by an auditory cue that predicts a reward, and you would still see a sequence of hippocampal activity. So what I and others in the field think that the hippocampus is doing more generally is creating a sequential representation of the order of events in our memories. And this might be anchored by the most salient events in those experiences, which in both of these cases is reward. So is the hippocampus, in addition to mapping space, creating a sequential representation of the order of events relative to where the reward is? And we know from previous literature, as well as work in our own lab, that this might be the case. So for instance, if you look at hundreds of place cells sorted here by their position of peak activity along this linear environment that the animal is traversing, and look at the reward location shown here with the white line, what you can see is that there's a clustering or over-representation, which Ashlyn introduced really nicely, of where these cells are firing, amplifying that reward location in this map. And recently, it's also been shown using 2-photon imaging techniques that there's a specific subpopulation of neurons that fire specifically at reward locations and will follow the reward when it moves, even while other place fields remain stable. But if the reward is anchoring an entire sequence of hippocampal activity spanning this whole experience, then we need to look more closely at what's happening out here, further away from the reward. And previous studies have largely assumed that this remapping is random out there, but we don't know for sure. So I hypothesize that if the hippocampus is anchored by reward, in addition to other stimulus features, then if you move the reward within an environment, you should see remapping of a subpopulation of cells to the same relative position with respect to reward. So for instance, not just firing directly at the reward location, but where this blue cell is firing before or this red cell is firing after. And this would be in addition to a stable spatial representation as well. And so we can test for this really robustly using virtual reality environments, which Alina introduced really nicely on Sunday, because you can tightly constrain the visual stimuli that the animal is receiving while also constraining their navigation path to making sure that they're actually sampling all the locations in the environment so that if there is remapping away from the reward, you can see it. Because otherwise, they'll just beeline to the reward and you might not actually sample that space. So we set out to do this. I collaborated with a really talented, now former graduate student in the lab, Mark Plitt, and we're doing 2-photon imaging of calcium activity in hippocampal area CA1. And so the way we do this is we head fix a mouse on a cylindrical treadmill here so it can run freely. It's surrounded by 3 monitors that deliver the VR environment or the video game. And then there's a lick port that can deliver a sucrose water reward when the animal licks. And unfortunately, to access the hippocampus, we do have to aspirate the overlying cortex. So as far as we know, this area doesn't have any lateralization. It's not the auditory cortex, but obviously we are thinking about that. And we express a genetically encoded calcium indicator here virally called GCAMP, which allows us to visualize the firing activity of neurons via changes in fluorescence so that we can record simultaneously from over 1,000 cells during behavior. And so in order to see if hippocampal activity is remapping relative to reward, we designed a task that has a virtual environment with multiple different changing hidden reward locations so that we have multiple opportunities to see this remapping. So this is a side view of the linear track. This is what it looks like from the front. So you can see the mouse kind of feels like it's running down a hallway here. And there's a 50-centimeter hidden reward zone that is, this blue shading is only for your eyes. The mouse can't see this. There's no discrete visual cue telling it where the reward is. It just has to learn through experience where it is and remember that location. And if it licks anywhere within this 50 centimeters, it'll get its reward delivered operantly. We trained the animal on this for a few days, and then in the middle of the third day, we switched the reward location to this purple location, for example, unbeknownst to the animal. The only thing that changes is where the reward is delivered. So now, by exploring the environment with their licking, they have to find the new location and remember it. And then we repeat this switch 2 more times. So now we have 3 possible opportunities to see this remapping within a single environment. And then to provide a comparison point for the remapping that we would see from spatially reorganizing the visual features, which is thought to be one of the primary drivers of hippocampal activity, we introduce a novel spatial environment that has a similar structure but different wall patterns and colors. And again, we have 3 reward location switches, and we reverse the sequence from the original, and we counterbalance these sequences across animals to control for the different positions on the track. So the goal of this task is for the animal to make moment-to-moment estimations of where the reward is based on its experience and its reward history, using the visual features that it has available to it and relying on its memory. And so all you really need to know about the behavior for today is that these mice can learn how to do this really well. So here I'm showing you the smooth licking in each row here as a histogram. And you can see when the animal is licking at the already learned reward zone, it's licking very precisely at the beginning of the zone because that's the first place that it can receive reward. And then when we move the reward location, they're able to adapt their licking really well over the course of the session. By the second time we do this switch, you can see how good the animal's licking has got for the new reward zone, and then again, they can switch really quickly. So the specific neural questions that I can ask with this behavioral data are first, how do place cells remap when reward is moved within an environment? What is the prevalence of any reward-induced remapping that we see? And importantly, do we see cells that remap to a consistent relative position from reward? So I'm going to show you 2 example mice here that experience these switches. These are individual neurons within each of those mice. So here I'm showing you trials or laps of this track on the y-axis, position on the track on the x-axis. The white vertical lines indicate the beginning of the reward zones, and the heat map is the firing rate of the neuron. So you can see that these are our classic stable place cells. They're firing the same spatial location across the entire session. Now remember that we're not changing any of the visual features when we move the reward within an environment, but still when we move the reward, we are surprised to see there are other cells with place fields that completely disappear. There are place fields that appear. There are cells that follow the reward location really closely, replicating some of that previous work that I told you about. And most interestingly to me, there were cells with fields further away from the reward, either after or before, or in this case, after and before, that also remapped really drastically. And you can see maybe in these 2 examples that maybe this is to the same relative location from the reward, but we're going to come back to that. So what this tells us so far is that moving the reward is sufficient to induce this neural remapping across the entire environment. And if we compare this to what we see when we change environments, we actually see a really similar collection of neural phenotypes. And so now just to quantify how prevalent this is across these 2 conditions, I'm going to compare days where we don't move the reward, which I'll call stay, and we just split the trials in half to have a pseudo before and after. Days where we switch the reward within environment and days where we change the environment. So each dot here is a mouse. Oops, sorry, my axis is gone. But you get the idea. This is a fraction of neurons. So this is 1 here. And we see an intermediate decrease in the fraction of neurons that have stable place fields from moving the reward within environment versus across, and an intermediate increase in the fraction of cells that remap. So what this tells us is that moving the reward in a constant environment gives us more remapping than we would from not moving the reward, but it's not a completely different representation. So the brain is still preserving this spatial representation of the environment, but signaling that something has changed. Okay, so when we move the reward, do we see cells that remap to that same relative distance? So to show you that, I'm going to take these same example neurons that I showed you before, and now all I'm going to do is for the trials following the switch, just align the reward locations. And so now what I hope you can appreciate is that all these fields line up quite well, and this is true even for fields that seem to be firing at the end of the track and then move to the beginning. And they did that in order to maintain their relative firing distance from that reward, which suggests that the hippocampus might actually be representing this repeated lap-by-lap experience of this track more as a circle. We also see this across environments. So these are cases where we are changing all of the visual stimuli, and yet we still see these cells locked more to where the reward is than those spatial features. Of course, this isn't true of all neurons because it's biology. So we also see cells that remap their fields to more random locations on the track. And so to ask if what I'll call this reward relative remapping is happening more than we expect by chance, we can take those aligned reward locations, put them at 0 on a circular axis, and now I can just measure, is there any difference in where these two fields are with respect to the reward? And so things at 0 indicate remapping to the same relative distance from reward, and so we see that indeed we have a much higher fraction of cells compared to a random remapping shuffle shown here with the red line. Interestingly, if I look at the last switch that the animals do at the end of this 2 weeks, there's a greater fraction of cells. And in fact, this increases linear across the task experience. So what this suggests is that the hippocampus is recruiting more neurons into this reward relative representation as the animal gains more experience with this task structure and learning that finding hidden rewards is the important thing to do in this task. So finally, if we put all of these reward relative neurons together, we can see that they make these really beautiful sequences that are dissociated from the spatial location on the track but are locked to the reward. So here I'm showing in each row is a mouse. I have 7 mice so far in this experiment. And within each mouse, I'm sorting the neurons that had significant reward relative remapping to their position relative to the reward before the switch, and then just maintaining that sort after the switch. So you can see that the sequences are really well preserved before versus after, and they span the entire environment, even though they're the densest around the reward. So this population is creating a sequential event order representation relative to the reward across the whole space. So to summarize what I've showed you, we found that moving the reward can induce this sequence of reward relative cells in the hippocampus in parallel to this stable spatial map, creating these 2 lines of information that are maintained together. And we see that these sequences increase in density around the reward location over experience, suggesting an increasing strength of this generalized task representation as the animal learns. So where I'd like to take this research in my future career is to ask more broadly, how do behavioral and physiological states shape the information that we retain in memory? So another way to say this is, how does the hippocampus know how to prioritize what to remember in any given context? And we can examine this on short time scales. So for instance, in the task that I showed you today, when we move the reward location, we induced a period of increased cognitive load on the animal. Now they have to stop using their memory of the previous reward location and learn a new thing and learn to remember this new location and hold these 2 things in competition and decide which one it is. And that increased cognitive demand induces this neural flexibility that we saw where we get this reward relative remapping. But we don't really understand how this is computed in the brain. Is it computed in other cortical areas and sent to the hippocampus? Is there neuromodulation at play directly into the hippocampus? And so these are some of the questions that I'd like to address. And at longer time scales, both physiologically and in my career, I'd like to move toward looking at how drastic changes in physiological state throughout life can have a profound impact on memory function. And one of the most striking examples of this to me is pregnancy and the postpartum period. Now, about 80% of pregnant people report memory deficits and changes in cognitive performance. And this is wild, and we kind of have no idea why. And despite decades of research on the hippocampus and establishing it as a center of memory and cognition and looking at place cells, we have very, very little idea what is happening to hippocampal physiology during pregnancy and the peripartum period. And so combining my experience with optical imaging and my PhD background, which is in electrophysiology and freely moving animals, I will assess how hippocampal coding properties are changing during pregnancy and this peripartum period to understand how those changes might contribute to the memory and cognitive changes that we see, and to understand what cognitive, hormonal, and physiological factors are driving these neural flexibility changes during this important period of life that affects so much of our population. So with that, I would like to heartily thank my wonderfully supportive advisor, Lisa, my amazing collaborator, Mark, and the whole Giacomo lab, past and present, my PhD advisor, Lauren Frank, for all of his support over the years, and my support team on my recently submitted K99, and then leading edge for being this incredible community that I'm so happy to now be a part of, and some of you for your help on the K99 as well. So yeah, thank you. Happy to take questions.
Beautiful work. Okay, questions? Yeah. Okay,
Very, very cool stuff. So I'm curious. I mean, you talk about this as a reward, and it makes sense that this is a reward because it's happening concomitant with you administering a reward. But how confident are you that this is not salience, or, I mean, would the same thing happen for punishment, or there are different punishment cells, that sort of thing?
Yeah, that's a fantastic question. So I don't think it is specific to reward. I think it might actually be more specific to the memory demand of remembering that location. And so actually one of my other projects in the lab is to, in freely moving animals, because that's easier to do than in the virtual reality, separate the location that they have to go to as their goal from the location where the reward is actually delivered to see which one of those it is. As for punishment, there are definitely cells in the hippocampus that respond to aversive stimuli, and this has been more thoroughly looked at actually in the ventral hippocampus, the bottom half. This is the dorsal hippocampus, that there are separate subpopulations of cells for rewarding versus aversive stimuli. So I would predict that we would also get an overrepresentation of an aversive thing, and maybe also see this relative remapping as well. And given different receptors that cells have, it's probably likely that they'd be at least partially non-overlapping populations, but we don't really know for sure. It's a great question, though.
Okay, finally, is there input to these cell populations specifically from, like, the ventral striatum? Is there something where we're encoding saliency out to the hippocampus?
So the ventral striatum does not project to the hippocampus, but the hippocampus projects to the ventral striatum, which is actually my PhD work. So yes, the dorsal hippocampus in particular is important for linking spatial information to reward-related or maybe saliency information in the accumbens, even more so than the ventral hippocampus, which I couldn't find any relationship between reward stuff with my ventral hippocampal recordings, which is a source of frustration. So yes, the circuit goes the other way, but I do think that the hippocampus is really well poised because it can create these sequences of activity, and it can send them out to downstream brain areas in like really fast plasticity conducive bursts that it can associate whatever it is encoding with things like saliency in other regions.
Huh.
Yeah.
Very cool.
So you had this like black and white graph with the little licks, and I noticed that on certain trials, after you switch, they'll just do this like long licking behavior for like ever before. Have you looked at anything specifically with that, and what it's kind of like they're probing? It looked like they knew where it was, and then they just do these random.
Yeah, it'll take me a minute to go back, but I might have it, on a backup slide that should have been closer to the actual talk. Sorry. Okay. Yeah, so there's two things happening here. So one thing is after the reward switches, you can see they lick in the old location for a few trials, and then they do this more exploratory licking. But these pink trials are actually omission trials. So I didn't mention this because I didn't look at it in this particular talk, but on 15% of the trials, we randomly omit the reward. That does a couple of things. It keeps them really engaged with the task, and it also allows us to compare times when they stop on the track and don't get reward to times when they do get reward, to kind of like Zara was asking, are there other features of this experience that could be driving it, such as the fact of just stopping or not moving? So when the reward is omitted, they do tend to lick down the rest of the track. The really interesting thing, though, is that they don't really do that before the switch. So they've gotten a pretty good idea of this probability estimate of, okay, the reward was omitted on this trial, but I think it's probably still going to be in the same location. So there's no point wasting the energy of continuing to lick. But after the switch, now that I've upended their world and they don't know what's happening, they will do this exploratory licking across the whole track, and then you can see it kind of start to condense back around the reward location as they gain that experience. So,
yeah, that totally answered it. Thank you.
So, Maeve from MSU. I know nothing about neurobiology. I was just like really interested when Zara brought up like punishment. Do you know if there's anything that does anything happen if you have punishment and reward at the same time for these types of events? Like, do they have to like weigh that in these, or like do you see firing of like both sets of cells?
Yeah, I have no idea in the hippocampus, but others have done that work, especially looking at like amygdala and other areas that are involved in the saliency and reward and punishment calculation. that would induce a really high cognitive load state, because now they're having to do this competitive calculation between, is it worth going and getting the foot shock or whatever it is to get my food? And I would imagine that that would sort of like amplify this whole representation, but we don't know. Yeah.
Really quick question. Do mice get mom brain? Like measurable
I don't
memory
know.
loss?
I need to find out.
Yeah.
There's very limited work. So in rats, they do have worse memory performance, especially in their third trimester, which is also akin to the worst memory performance in humans. They actually tend to have slightly better memory performance earlier in pregnancy. And then during the third trimester and early postpartum, it's really bad. And that's even if you control for like how much they're moving. It's not just that they're uncomfortable, like they have worse spatial memory. And then following birth, there's increased neuroplasticity, increased neurogenesis in the hippocampus, and they tend to outperform rats that haven't been pregnant. So there's really dynamic changes, but whether in this kind of task I would see deficits, I don't know yet. That's a future thing to figure out.
I also have a quick question. So it looked like the second time you moved the reward location, the mice, at least from what I could see kind of subjectively on the graph, the mice learned faster. Like they updated their licking behavior faster than the first time that you had moved the reward location. So did the place cells remap in a corresponding time, or do they remap, like what's the relationship of the remapping to the behavioral change?
Yeah, that's a great question. That's kind of like immediately next on my to-do list of analyses. There's definitely variability in when I see the cells remap, and I would predict that it corresponds to when some certainty threshold has been crossed of, okay, now I estimate that the reward is going to be here, and so it should predict the change in licking behavior, or maybe vice versa. Maybe the licking behavior happens before the neural change. So I'm looking at that next. Stay tuned.
Cool. Okay. Thank you.